I'm not ready to get back to the black sea or like all kinds of things. I don't know, brain.
Think we're gonna get into depth with that?
No, I don't know. I haven't looked at the slides yet.
Good try.
My laptop was dead.
Yes. This one's just like.
Yeah. No one knew what a state of care.
I got that.
Wearing white for fishing. This is like a sweatshirt. Oh, is this for your, for the summer? Oh.
Pretty much like buy it now.
That's fine, 6. She's each way. It's not terrible for Alaska.
They reimbursed me 7. This one in there.
Oh, nice. That'd be nice to go, but.
Alrighty, good morning, everybody. How are you all doing today? Yeah, midway, tired. Alright, I'll take quick attendance. Tegan? Lucia? Nicolo? Autumn? Ryan? Quinn? Celeste? Zoe? Annika? Jesse? Sarah? Kata? Clayton? Paige? Erica? Victoria? Brooke? Abby? Hannah? Here. Gabby? Here. Juliana? Nicholas? Henry?
Here.
And Tyler. Awesome. Richie, I have your exam. All right, so today we're going to be kind of diverting back to bacteria. So lecture 6 was all about viruses, both eukaryotic viruses and bacteriophage. And so today we're going to be switching back over to bacteria. And you know, these cells have these really tiny machines that are able to you know, do transcription and translation in order to make an RNA message from DNA and then decode that RNA message to make a functional protein. So here are the suggested readings for this part of, or for this lecture. So I will be also continuing on with this, this transcription translation stuff next week on Monday. So today we're kind of just going to go through half of the chapter. And so an overview, so whatever is bolded are the things that we'll be talking about today, and then the things not bolded will be what we'll go through next week. So first we'll start with talking about RNA polymerases and these sigma factors that they have that essentially tell the RNA polymerase where to go and which genes to transcribe. Then we're going to talk about how transcription works, how you go from DNA to RNA, and then we'll do some Bio 101. We'll talk about the genetic code and ribosomes and transfer RNAs or tRNAs, and then lastly we'll talk about translation. So some of the machinery necessary for the proteins have to be folded and post-translationally modified, and so these proteins can then be degraded or exported, and we'll talk about that all next week. So today we'll go through the basic overview and get some nomenclature down for you guys. So as a quick introduction, the cell has this really vast genome, and it can access its genomic DNA through two ways. The first is transcription and then translation. So in transcription, that is when you have a DNA template that is read to make an RNA copy, and the enzyme that does this is an RNA polymerase. Translation, you have the mRNA transcript, and that is then translated by this ribosome, this really massive RNA protein machine that can synthesize the polypeptide sequences from amino acids in order to make a protein. And then after translation, each polypeptide has to be properly folded and placed at the correct place in the cell, either intra or extracellularly. And then when these proteins have outlived their usefulness, they can be destroyed, and those amino acids can then be recycled. So let's hone in a little bit more on transcription and what these RNA polymerases and sigma factors do. So RNA polymerases are these complex enzymes that carry out transcription by making copies of the RNA, which we call transcripts, from a DNA template. And in bacteria, this RNA polymerase is this holoenzyme that we call, and the reason we call it this is because it has to have two components together. You need to have the core polymerase structure and the sigma factor attached. So the core polymerase consists of 4 components. There's an alpha 1, an alpha 2, a beta, and a beta prime. And so these 4 components, and I'll show you a picture on the next slide, are required for initiation and elongation of transcription. And we'll go through a little bit more of what those steps are. Then there's the sigma factor that is needed in conjunction with this core polymerase, and this is required for transcription to be initiated. So it's required only for the initiation phase and not for elongation. So there's a whole bunch of different types of sigma factors in bacteria, and they are really important because they tell the RNA polymerase which gene set to transcribe given whatever environmental conditions the bacteria are in. So if you're in really low nutrients, the sigma factor can tell the polymerase then, for instance, to make more flagella so that they can get out and search for more nutrients. So the sigma factor is important because it provides the specificity of which gene sets the bacteria has to turn on under a given condition. And so the sigma factor just basically places the RNA polymerase at the right site for transcription, so it's only needed for initiation. So there are these 5 subunits. So basically the core polymerase consisted of these 4 subunits, and then the sigma factor is just sigma. So these 5 subunits make up this holoenzyme. So just to give you a little bit of background, so over here in red we have this sigma factor. It's this is the sigma sign in Greek, and so over here we have the DNA channel, so this is where the DNA will want to bind, and so we have our alphas and then our betas as well here, the beta and the beta prime. And so if you kind of turn it around on its side, you can see that the other alpha is over here. And so basically the thing that's important here is that the alphas, the alpha 1 and the alpha 2 are kind of just like hanging out on the sides. They're not really in the center where the DNA channel will be going in. That's where the beta prime is. So the alphas are more structural. They just provide more structure to this RNA polymerase protein. And so this sigma factor here in red basically loads onto this RNA polymerase, so it kind of attaches over here, and so where this dashed line is shown, if you guys can see that. So that's important because it's loading right where you have this catalytic site. So the catalytic activity or the enzymatic activity of the RNA polymerase is really in the center of this entire protein structure. So this beta prime is what's housing this catalytic site for RNA synthesis. So that's an important thing to know, that the catalytic site really is the beta prime. So the sigma factor is needed because it basically has to bind into this RNA polymerase to create this holoenzyme, and that helps the RNA polymerase then recognize the promoter, which is the site where transcription will begin. So what is a promoter? So a sigma factor, like I said, first will bind the polymerase to create that holoenzyme, and then it will help the polymerase detect a specific DNA sequence that we call the promoter. The promoter basically tells the RNA polymerase where to start making the transcript. So it's the beginning of an open reading frame. So in this picture right here, it's just the very beginning of this sequence. So there are some genes that have to be turned on all the time. And so basically here, well, I guess let's start with this. So a single bacterial species can make several different sigma factors. And I said this because bacteria are always in this fight or flight or feast or famine environmental niches where they have to be making different things all the time to be able to survive. So they can have different sigma factors. And so every cell has a housekeeping sigma factor. And so what that means is there are certain genes in bacteria that have to be turned on all the time. And so there's a sigma factor that bacteria have that's called the housekeeping sigma factor that basically just keeps the light on. It just makes sure that all of the genes that are needed to be transcribed are turned on. So a housekeeping sigma factor is just one that the bacteria will always have. It's always going to be on just to make sure that bacteria is able to survive. But there are many, many different sigma factors that a bacteria can make. Just as a note, you don't have to remember this, but in E. coli it's just called sigma 70. So housekeeping genes are just genes that are always going to be expressed. We use them as controls in science and in research, so they're really important. So you can get transcripts here that are monocystronic. And so basically what this means is you have a promoter upstream of just one gene. And so therefore transcription will give you an mRNA transcript of just that one gene. But what's cool about bacteria is that you can have a promoter that drives transcription of a gene set. So here we have gene 1, 2, and 3 that make up this gene set. And so usually these gene sets have a related function. So the transcript that's made here is called polycystronic RNA. So poly meaning many, mono meaning single or one. Yes.
So just to check, the monocystronic means like it's going to turn on just one gene, and then the polycystronic is multiple?
Yeah, exactly. So when you have polycystronic RNA, basically this is a promoter right here, and so the transcription will occur and you make this RNA. And so this RNA is a 3-gene RNA transcript. And usually they are made together because they all have related functions to turn something on. So say to make flagella, you need 3 genes to make the flagella. normally you'll find them all on the same place on the chromosomal DNA, and so that they usually will require one like sigma factor that recognizes this promoter sequence, and they're able to be transcribed. So yeah, exactly. Does that make sense? Any other questions on this? All right. So, like I've said, the sigma factor helps the cell readjust its physiology. So the cell has to constantly adjust its gene expression profile in order to survive. So different gene sets will have to be turned on or turned off. And so basically this is largely done by these sigma factors in order to deal with these different conditions or these changing conditions. So bacteria, like I said, they wander in and out of stressful environments, and so they have to turn on certain genes to do that. One of them being like flagella, for instance, to help them move and search for other things. So their different gene sets are controlled by different sigma factors, and some of these changing environments can be like heat stress. If there's starvation in the environment, there aren't enough nutrients, basically the cell can then turn on sigma factors that will express some sporulation genes, some genes to create spores to help it kind of remain quiescent and latent and, you know, can hide out, hibernate until there are plentiful nutrients for it to start growing again. Biofilm formation is also another example. So this kind of is connecting some of the things that we've talked about in the previous, the first unit. So now we're kind of zooming in a little bit more as to why and how things happen, and so why bacteria are able to switch so quickly in changing environments. It's these sigma factors that are really important, and there are so many different ones that each bacteria has. So quick concept check. The sigma factor is directly required for what? Transcription initiation, elongation, translation initiation, or elongation. We haven't even talked about translation yet, so which one? A. A. Nice. Super easy, but just want to make sure you guys know that one. Okay, so now we're going to talk about the 3 phases of transcription. There are, so initiation, we talked about a little bit. We'll talk about it a little bit more. There's elongation and termination. So in initiation, you have the RNA polymerase as the holoenzyme, so it has that sigma factor present, and that way it's able to bind to the promoter. This is then followed by melting open of the DNA helix and allowing synthesis of the first nucleotide of the mRNA. In elongation, you have this sequential addition of ribonucleic triphosphates or RNTPs. That's just the genetic code essentially for RNA, and these sequential addition of this RNA chain is usually extended at the 3' hydroxyl end, so it's the 3' end of the growing mRNA chain. And then during termination, there are sequences at the end of a gene that trigger the release of the polymerase as well as the RNA transcript. So the RNA polymerase will detach from the DNA after the transcript is made, and there's some sequences that help trigger that. That's just a little quick intro, but now we're going to go a little bit in depth into all these steps. So over here, we have this RNA polymerase that is this holoenzyme. So you see you have the red sigma factor, and then you have the 4 other core RNA polymerase units here, the 2 alphas and the 2 betas. And this beta prime is that huge chunk of the RNA polymerase. Remember, that's the one that carries the catalytic activity, the enzymatic activity. And so together as a complex, this is loosely bound onto the DNA. And so basically, it's loosely bound because it's then able to scan the DNA. It's not bound really tightly. So it can scan the DNA to allow it to find the promoter sequence. So the promoter sequence is right here. It's jumping around and it's scanning. It's loosely bound. And so once we get this binding to the promoter sequence right here, you get this conformational change from the closed complex to like this open complex. So basically, from the open complex where it's loosely bound to this closed complex. So once it finds the promoter, you get this conformational change and it kind of closes onto that DNA sequence and is able to then move on. So, and then once it attaches and you get this closed complex, now the RNA polymerase can start to unwind the DNA and begin transcription and starts to, add the DNA or the RNTPs. And so at this point, it starts to make the first part of the mRNA transcript, and then the sigma factor now realizes it kind of stays until about 9 base pairs have been joined, and then it knows like, okay, I've done my job, I got the RNA polymerase to the promoter site, and now I can leave because it's not needed, remember, for elongation, it's just needed for initiation, and so it knows to just kind of dissociate when the RNA polymerase starts to make this, this transcript. Does that make sense? So that's initiation in a nutshell, and now elongation in another nutshell is after that first base is placed, you start to have elongation of this chain, this sequential addition of these ribonucleotides, which we call RNTPs. So now the RNA polymerase, without the sigma factor, because now we're on elongation and it's not needed, it continues to move along the template and it synthesizes the RNA, and then at this point it does it really, really, really fast. About 45 bases per second. So remember, bases are the A's, C's, T's, and G's. And so those are what we're talking about there. In this case, it's RNA. So as the RNA polymerase is synthesizing the transcript, basically it requires the DNA to unwind, right? And so the unwinding of the DNA ahead of the moving complex of this RNA polymerase starts to form a transcription bubble. And that transcription bubble causes some supercoiling. The DNA just starts to supercoil ahead of where the transcript is being made. And so, just as a note, there are DNA topoisomerases, which basically relieve the supercoils that are upstream of that transcription bubble just to help relax the supercoiled molecules. But the important thing to know here is that elongation is the sequential addition of these RNTPs, and the RNA polymerase doesn't have the sigma factor. And so it just moves along the template and it synthesizes these RNA molecules at a really, really fast rate. Make sense? Okay. So that's elongation in a nutshell as well. So now we're going to talk a little bit more about transcription termination. So how does transcription know when to be terminated? So the RNA polymerase knows when to stop or where to stop elongation in bacteria in 2 different ways. So the first way is row dependent. Basically it relies on a protein called Rho, hence the name. And so this creates this like really strong pause site that the RNA polymerase it basically sees, and then it knows that, okay, we need to stop. And I'll talk about this on the next slide. There's a row independent termination pathway. Basically, here it doesn't require row, hence the name. And so it requires the formation of a secondary structure on the RNA. And so a really rich GC region on the RNA, and then these 4 to 8 consecutive uridine residues, I'll talk about this a little bit more, that basically helps to weaken the association and allow the RNA DNA association, and it allows the transcript to pop off. So we'll talk a little bit about this. So in this process, in the row dependent termination process, let me orient you here a little bit. So we have the DNA here, going through, and the RNA polymerase is all right here. And so the movement of the polymerase, it's going down this way. So this right here is a pause site that it's recognizing. And then this right here in yellow is this rho protein. So basically here, what happens is this rho protein that's shown in yellow is able to bind this RNA transcript that's being made. And what it kind of does is, if you can imagine, is it's pulling itself closer towards this pause site. So it literally like uses this RNA transcript as it's being made, and it pulls itself up towards this pause site, and towards the RNA polymerase. And so once there's contact between this rho protein and the RNA polymerase, that causes termination. So once the rho touches the polymerase, it has this helicase activity that can rip the RNA DNA hybrids, and it pauses or terminates transcription. So for the rho independent termination here, so basically here, if you kind of want to orient you here again, so you have your DNA here, this is the transcript, the RNA transcript being made, your RNA polymerase. If we zoom into this little section right here at this pause site, you have this opening of the DNA because it's being transcribed, right? So basically, there's this GC-rich region that forms this hairpin loop. So this is a really rich GCs, consecutively, and that causes this hairpin loop. And so basically, this GC-rich area, as well as this NUS-A protein in orange here, both of these help to stabilize this hairpin loop and just kind of keep it stable as this hairpin loop. And so this basically, the NUS-A and this GC-rich area are essentially what stabilizes the stem loop structure really well, and basically will pause the RNA polymerase on the transcript. So now the RNA polymerase can't go anywhere because you have this essentially like a knot in the DNA, and it just can't go further anymore. And so this NUS-A protein in orange has to be there in order for this to happen, and there's got to be contact between the hairpin loop, the NUS protein, and the RNA polymerase in order for termination to occur. And so the next part of this mechanism that I want to talk about right here is this UA region. So this is a uridine-rich region. It's just downstream of the stem loop. This forms this poly-U site. It's a stretch of uridine, and basically what it does is it weakens the DNA, so there's really weak associations between the RNA transcript and the DNA template. And so just by pausing this polymerase, basically this mechanism just wants to pop off, because now we have this hairpin loop structure, this really weak association here, and so it just wants to pop off. So this is what allows transcription to pause and be terminated without the need of the rho protein. So some things I want you guys to know are what causes termination in both of these different scenarios. Any questions on that? Okay. So there are several RNA types, each with a different purpose. So up until now, I focused on mRNA or messenger RNA. And so basically, these are the transcripts, which are the messengers to make proteins by the use of ribosomes. But what I'm showing you in this chart is that there are several other classes of RNA, and each is designed for a different purpose in the cell. So rRNA right here, or ribosomal RNA, hence the name, forms the majority of the ribosome. The ribosome basically synthesizes proteins. So the rRNA is literally, the ribosome is like mostly rRNA, just some protein is in it, and I'll talk about that more. We also have tRNA or transfer RNA, and basically that shuttles the amino acids to the ribosome to synthesize a polypeptide sequence. So the tRNA is kind of this like adapter molecule that brings the amino acids to the ribosome for it to be able to make proteins. There are also these sRNAs, which are called small RNAs or regulatory RNAs, and so these don't decode a protein, but they can directly regulate transcription and translation. We're not going to get into those today. There are also these other tmRNAs, which are pretty unique. So all you have to know is that their function is to strip off ribosomes from damaged mRNA. They free the ribosomes that are stuck on damaged RNA. And then finally, there's catalytic RNA. And so these have enzymatic functions. So catalytic and enzymatic are synonymous. So these basically will have an enzymatic function, and an RNA with an enzymatic function is called a ribozyme. So we'll talk about that more. And then one thing I want you to notice is this dramatic difference between their half-lives. So mRNA is made, and the half-life essentially is the length of time that that molecule degrades half of itself. So the average half-life for mRNA is about 3 to 5 minutes here, but then there's these ribosomal RNAs that their half-life is hours, as well as this tRNA, and so we'll just note this, and this will make sense a little bit more throughout the lecture as to why there are some differences in their half-life. So yeah, the average half-life for mRNA is about 3 to 5 minutes. Some are 15 seconds, some can be 30 minutes. But this basically changes or indicates the stability of the different kinds of RNA. So the stability of the different kinds of RNA differ drastically. So like I said, the tRNA and rRNAs are much more stable because they have a longer half-life. So longer half-life means that they're more stable. And that's going to be important for the cell. And so the ones that are more stable, they are more stable because they have modified bases that are less susceptible to RNA digestion. And we'll talk a little bit more about that as well. Any questions on this? All right, so a quick concept check. Termination of transcription can be caused by which of the following? B is right. Wait, sorry, did you say B?
D.
D, yes, exactly. D is correct. So you can have the row independent stem loop structure that forms from the GC-rich residues, as well as the row factor is another way, and the row dependent. We haven't talked about stop codons. So yeah, so A and D are correct. So to quickly go through some more Bio 101 here, once a GNA or gene has been copied into mRNA, the next stage is translation. So we're decoding the mRNA to synthesize the protein. So an mRNA molecule can be thought of as a sentence in which you have triplets of nucleotides that we call codons. And those codons represent individual words. Those words can be amino acids in theory. And that final sentence that you make is the polypeptide sequence. And so ribosomes are the machines that read the mRNA language and translate it into protein. And so they do this via the genetic code. I'm sure you've probably seen this at some point in your careers. So the genetic code consists of nucleotide triplets called codons. So 3 nucleotides make up a codon. There are 64 possible codons. I'll show you a picture of all of them. You don't have to memorize them all, but what you need to know is that 61 of them specify amino acids. Within these 61, that includes the start codons, or the start codon, and then there are 3 additional stop codons that make 64 possible codons. And then the genetic code is really redundant or degenerate, so multiple codons can encode the same amino acid. And what's really cool here is that this code, this genetic code, operates universally across species. So across all 3 domains of life, we use the same exact genetic code. And basically this is applicable to all of life with very, very, very few exceptions. So whether you're a human or you're a phage, we all use the same genetic code. So that's actually pretty like marvelous if you think about it. And so here, this is the genetic code. And so codons within a single box will encode for the same amino acid. So over here you have all of these valine, or like these glycines right here. Basically, glycine can be translated by 4 different codons, and usually you'll notice they differ in just the last base only. And so you don't need to memorize all of these, but one that I do want you to know is this methionine or MET. It's this AUG sequence. This is the start codon for all proteins in bacteria. It's always going to be the start codon, this methionine. And there's only one way to make it, so there's only one codon for methionine. In red, we have these 3 stop codons. You can see they're also very similar, differing just a little bit. Any questions on this? So before the ribosome can start translating the mRNA, there's this adapter molecule, the tRNA molecule. So this stands for transfer RNA. And so it's this decoder molecule that converts the language of RNA into that of protein so that the ribosome can recognize that language and be able to make the protein. So basically they convert the codon alphabet of RNA into an amino acid alphabet for proteins, because the ribosome is going to be recognizing the amino acids. So these tRNAs, so this is the tRNA right here, this is the mRNA sequence, this is the AUG, this is the start codon, the methionine, and so here's an amino acid that it's recognizing. And so these tRNAs are really just floating around in the cell, all around the cell. And so they have to get charged with the right amino acid, and then they are able to return to the ribosome and drop off that amino acid cargo. And this is achieved through 3 functional regions of the tRNA. The first is this anticodon. Basically, this is responsible for hydrogen bonding with the mRNA codon that specifies the amino acid. So can you guys take a guess? I kind of already alluded to this, but what does the anticodon specify for? Which amino acid? So this is the anticodon, and it's specific. So it specifies for what? The start codon, exactly. So this anticodon is what has to come in and find the RNA to be able to know, okay, this is the start spot, and so it recognizes this methionine and it hydrogen bonds there. And then there is the 3' acceptor end. So this right here is a 3' acceptor end, and basically it's where it's accepting or binding the amino acid. That's the part that is charged with the right amino acid that is matched to that particular anticodon. All tRNA molecules will have this 3' end with CCA, so this is the acceptor end. And I just realized this morning, like CCA or ACC, you can remember that because acceptor starts with ACC. I don't know if that was meant to be that way, but just something I noticed, a good way to like remember it. So this is where this tRNA molecule gets charged. It needs to have this or CCA, and then it gets charged by the amino acid at the acceptor end. So the last thing here are these side loops. So that's these loops right here. They have these interesting Greek letters. Don't know what they mean. You don't need to know that either. But basically, what this does is it modifies the bases and creates these like really weird, like there's D's in there and this like Greek letter in here. And so the reason for these side loops is they create these side loops so that it causes this molecule to become more stable. So it just becomes more stable because there are just differences in the sequence here, and it's creating this loop, so it's harder to like break apart and move. And so, yeah, so these side loops are recognized by enzymes that match the tRNA to the proper amino acid. So they're important to keep this tRNA molecule stable for recognition of the amino acids. And so, like I said, tRNA molecules contain many unusual modified bases that are there because they help to protect them from being degraded by RNases. So these create more stability, and therefore, this is a reason why the tRNA molecule has a higher half-life or longer half-life. That's the connection to the previous slide where I was talking about tRNAs having longer half-lives than the mRNA molecule, because the mRNA molecule doesn't create these really stable loops where you get these modified bases. Does that make sense? So once the tRNA molecule does this and gets charged with the amino acids, it brings the that amino acid sequence to the ribosome, and the ribosome then can convert the mRNA code into the amino acid sequences for the proteins. So the tRNAs are charged, they're floating in the cytoplasm, and now they can bring the amino acids to the ribosome to convert the mRNA code. And so ribosomes are composed of two subunits, and so the two subunits are some rRNA or ribosomal RNA and proteins. And so in prokaryotes, the subunits are 30S and 50S, and they combine on the mRNA transcripts to form the 70S ribosome. So note here, so basically the ribosome consists of this small subunit plus this large subunit, 30S and 50S. So you'll notice here that 30 plus 50 is not 70, and so this is not an additive thing, so just so you know, as like an FYI, this S is actually a unit of Svedberg's, and basically what this is like a rate in which the ribosome subunit can sediment under centrifugal force. So you don't really need to know much about that, but just it's not additive, and so that's why it's 70S. But these two together have to come together to make the ribosome. So the ribosome, think of ribosomes as a ribozyme, which is basically an RNA molecule that has enzymatic activity. And so this enzymatic activity that the ribosome has is called the peptidyl transferase. So that just means that it can make a peptide bond that's necessary to stitch the amino acids together to form the polypeptide. So the peptidyl transferase is this enzymatic activity that the ribosome has, so we basically call it this ribozyme, an RNA molecule that carries out catalytic activity. This peptidyl transferase is part of the 50S large ribosomal subunit. And so just to give you a little bit more context here, we have in blue the rRNA, and in yellow we have protein. This all makes up the ribosome. So remember I said ribosomes, you can think of it as a ribozyme, and the reason is because these ribosomal proteins don't do anything functionally for the cell, they're just there to add structure, or not to the cell, to the ribosome. So the proteins add structure to the ribosome. It's really the rRNA that is creating this enzymatic activity and doing the work. So this catalytic center, usually the area where there's enzymatic activity is in the very center of this molecule, and you can notice that it's all blue, so it's all this rRNA. Okay, so basically, given the importance of rRNA in translation in bacteria, as you can imagine, the sequences of this is really like similar among a lot of bacteria, and so we'll talk a little bit more about that. But essentially, what you really should know here is that the ribosome is made up of rRNA and protein, mostly rRNA, and that's the part that's carrying out this catalytic activity. And so basically, it's really important to translate proteins properly. So this is usually conserved among a lot of different bacteria. There are very minor differences in the rRNA sequences. So basically, when you have differences in certain ribosomal rRNA sequences, that increases in basically the differences increase in relation to the evolutionary distance between species. So another way to put that is, if basically I want to identify an organism and see whether it relates to another bacterial species or organism. What we do is, if you've heard of 16S rRNA sequencing, so the 16S region is conserved among all of bacteria, and so if I have an unknown species, I can use this 16S region to try to sequence what that transcript is, and then work backwards and get the DNA sequence from that mRNA transcript, and then you can be able to tell if that relates to another bacteria that we know its sequence. So rRNA basically serves as this molecular clock for evolutionary biologists, because it's a way to measure the time in which two species have diverged from one another. So that's why this rRNA is very conserved among bacteria. When there are changes, it means that there's been a change in the last common ancestor between the most recent common ancestor between the different types of species. So the RNA really serves as like a molecular clock for a lot of molecular biologists. Does that make sense? All right. So the 70S ribosome, the whole entire ribosome structure that consists of the 30 and the 50S, has 3 binding sites for the tRNA. So there's an acceptor site that basically binds the incoming aminoacyl tRNA. So basically this is just the charged tRNA molecule because it has an amino acid bonded to the tRNA. So this is what we refer to as the charged tRNA. So that's the acceptor site. There's this P or peptidyl tRNA site. Basically this is the site where the tRNA is harbored and you can grow that polypeptide chain. And then there's an exit site that binds a tRNA molecule that has been recently stripped of its polypeptide or the amino acids. So once the tRNA is no longer charged, it's in the exit site and it knows that it needs to leave. Yeah, so I'll talk a little bit more about this. So this is a picture of a ribosome. And so the mRNA here is in this light blue color, 5' end and 3' end right there. And so in red, you have this 50S subunit. In this pink, you have the 30S subunit. And you have the polypeptide sequence that's being made here. So in the center, if you pay attention, this is the catalytic site. And so in blue, this blue center is the acceptor site. So that's the tRNA A site where the tRNA can bind. P here in green, the tRNA P site is where the polypeptide chain grows. And then here we have the E site in yellow, and that is where the tRNA can exit. So the tRNA really binds and does its thing at this very core center of the ribosome. So that's the catalytic center. So kind of bringing this back full circle. In the first couple lectures, I talked about how bacterial transcription and translation are different than eukaryotes because bacterial transcription and translation are coupled, which means that since bacteria don't have a nucleus, the ribosome can immediately bind the 5' end of the mRNA transcript that's being made, even before the mRNA polymerases are finished making the mRNA transcript. So the simultaneous binding of both the mRNA and the protein is called this coupled transcription and translation here. And so eukaryotic microbes use the separate cellular compartments to carry out most of their functions, like transcription, because the DNA is in the nucleus, so you're getting transcription happening in the nucleus, and then translation in the cytoplasm, whereas in bacterial cells, you have it all happening within the cytoplasm. And so just as another note here, another name for this coupled transcription and translation is co-transcriptional translation. Okay, so once a ribosome begins translating the mRNA, another ribosome can immediately jump onto the ribosome binding site. So in this TEM image, you can see here that you have, it's really difficult to see, but this right here, this like clothesline thread, that's the mRNA. And so you have your RNA polymerase here and these ribosomes that are trying to make the amino acids. So each one of those dark circles is a ribosome. And so you can have more than one ribosome jumping on to a ribosome binding site in order to make more protein. And so therefore the mRNA can have many ribosomes moving along its length at the same time, and this multi-ribosome structure is known as a polysome or a polyribosome. Usually they're closely packaged along the mRNA, kind of like you see here. And the reason being is it provides more stability once again, and so it protects the mRNA from RNases and helps speed the production of a single transcript. So this is useful because they rapidly translate proteins under that particular condition. And so having many ribosomes on the transcript helps to protect its stability. So if we go back to this image here, so this is just what I had on the previous slide, we saw that the rRNA and the tRNA have an approximate half-life of like hours. And so that's why they have these differences in their sequences, and they basically have more stability because they have higher half-life. So if they're more stable, they're not going to be degraded as quickly. So can anybody tell me why we don't have more stable mRNA molecules, or we don't really want the mRNA molecule to be super stable and have these really unique residues or bases, and so their half-life is a lot smaller or shorter?
Maybe mutations get a cure.
Yeah, that's a good point, yeah. So yeah, basically, remember we said that the environmental condition for bacteria changes really, really rapidly. And so the bacteria has to be ready on a dime to switch its genes and be able to make new things for it to be able to survive. That step happens at the mRNA, from the DNA to mRNA. So that's why the mRNA is not really as stable, and so it kind of gets degraded really quickly because the bacteria is ready to make more mRNA and make more proteins. But we need the protein part with the tRNA and the rRNA for the ribosome to be more stable, because once the bacteria has made its mRNA and for those proteins, then you kind of want more functionality and stability at those steps. But since the bacteria has to be able to change on a dime and switch its code really quickly in order to survive certain conditions, that is why the mRNA has a really low, a short half-life. Does that kind of make sense? Okay. So, this is important for next lecture, so I really want you guys to kind of look through these notes before next lecture. Next lecture, I will have more on this, just talking about how proteins fold and how they're degraded and transported in the cell. But it's important to know these concepts from this lecture for next week in order to really understand it. So I'm going to leave you guys with this here. This is kind of the background, and then we can talk more about function next week. So, questions?